All right. So in chapter 2 in Mary Van Hove's Anatomy and Physiology book, it covers some basic principles of chemistry that you need to understand. Now, again, bear in mind that this isn't a chemistry class. We're not going to go over obviously all aspects of it, but there are some important concepts and aspects of chemistry that do relate to biology and physiology that we do need to know, because that's realistically what physiology is. It's a combination of cell biology, chemistry, and physics that are applied to the functionality of the human body. All right, and as I've mentioned before, I myself am not a humongous fan of chemistry, but that doesn't mean it's not important to know. Um, I always think of the movie Quigley Down Under, if you guys have ever seen that before. And it's kind of an old Western movie that takes place down in Australia with Tom Selleck. And throughout the entire movie, everyone's trying to give him a revolver, pistol to use throughout the movie. And he keeps saying, I just don't have a use for one. I don't have a use for one. And and then at the end of the movie, that comes down where he has to, like a typical Western movie, there's a shootout. Him, they're going to try to draw as fast as they can, him and a couple other guys. And, he whips the pistol out and mows all three down. And he walks up at the end and he says to the guys he just shot, and he said, just because I said I don't have a use for one doesn't mean I don't know how to use it. Okay, so just because he didn't much care for one didn't mean it wasn't an important tool for him to use at some point in time. I mean, obviously in the movie scenario, that was a survival thing. For here, this is for, obviously your academic and physiology knowledge here. So, I don't know, I just always think of that movie when I bring that up, so I figured I'd share that. So, when we go through this chapter and we talk about some specific topics, a couple of things that we're going to cover in this, I'm going to start out just by talking about the basics of matter and atoms, and how they interact via chemical bonds. There's a few important characteristics, basic characteristics you have to know about atoms and how they form chemical bonds with one another, because you're going to be learning about, especially when you really start learning about human metabolism and how that all works. I mean, that's all basic applied chemistry. I mean, at this level anyways. I mean, obviously, if you go higher up, it's a lot more advanced. Obviously, I mean, water, that's important to know and understand, because what are we primarily made out of? Water. Okay, that's a big part of our composition. And understanding how water behaves, and the general chemistry of water is very vital for you guys, because that's going to help you understand, how substances are transported in the blood. Okay, how medications are circulated, how absorption of nutrients may take place, how we regulate salts and water levels in the body. I mean, there's a lot that goes into understanding water. There's a lot of important information that's really going to make a lot of sense with a basic understanding of water. And then pH, because pH is important to know and understand, just because this is a big part of keeping our metabolism in check, making sure we keep, or I should say, keeping it running properly, because if we don't keep our pH in check, our metabolism is going to go way off kilter and it's going to kill us. So we have to make sure that we're maintaining the proper pH. And then the last part of this chapter is going to consist about just the basic knowledge of organic and biochemistry, basically just talking about carbon-based molecules like carbs, fats, proteins, and nucleic acids, and how we use them in our everyday life. Their composition as well. And this is important to know and understand, because, when we're going into cell structure and transportation across cell membranes, or the structure of cell membranes, there's a lot of organic and biochemistry in there. And from a nutritional standpoint as well, it's important to know and understand what, these molecules consist of, and the different forms of, carbs, fats, and proteins out there, because when you get into more of a specific nutrition class, or your focus when you're trying to do with meal planning or certain nutrients that people need, you have to know what people are consuming so you can get the right fuels and molecules inside yourself. So these are the basic, the major core, the major concepts we're going to cover in this chapter. And so let's start out by talking about matter. Matter is anything that has mass and takes up space. So basically any physical presence out there in space that, that you can essentially measure or see. Okay. Right now, your computer has mass. You have mass. Your phone has mass. Your dog or cat has mass. The air is consists of matter. I mean, you can feel the air hitting you, okay, when the wind blows. You know that there's humidity in the air. There are gases in the air. So even air is matter, okay? And just because you cannot see all aspects of matter doesn't mean it doesn't exist, okay? And we typically find matter in three different states. This is a solid, liquid, or a gas, okay? And matter can move, back and forth between these three, okay? And the biggest part, or the biggest factor that will determine this, is temperature, okay? Obviously, the warmer the temperature, the closer the matter is going to get to becoming a gas. Okay, because because remember that matter, the smallest units of matter, which I'll talk more in depth about in a little bit, are atoms. Okay, those little things that have protons, neutrons, and electrons. Okay, and the higher the temperature, the more the energy is for these atoms to move around and spread apart. Then these, molecules, atoms, are going to have more space in between them. So as a result, you'll develop liquids and gases. Okay, so the cooler temperature gets, the closer you get to a solid state. Okay. And this is important to understand. For example, when you get into the respiratory system and we start talking about proteins and hemoglobin, okay, as we're going to learn about later in the chapter, proteins are these kind of big coiled, well, I mean, it depends on the shape you find them in, but hemoglobin is a big coiled protein you find on red blood cells. And so think about this. This is made up of a certain group of molecules, and we use hemoglobin for. We use hemoglobin for circulating oxygen around. It's part of our red blood cells, okay? And oxygen kind of sticks to the center of it, and an iron there. And if our body temperature gets too warm, these proteins may uncoil. Oh, whoops. Sorry. These proteins may uncoil, and then as these proteins uncoil, that may cause the hemoglobin molecule to unload oxygen because, again, the increased temperature, this is going to become less of a solid, okay? And vice versa, if it gets too cold, okay, if our body temperature gets too low, these proteins may coil, too tightly together, and we'll have a harder time unloading oxygen, okay? So the temperature of our body will affect certain states of matter, I mean, within us, and this is just one of many examples that we can discuss, okay? I mean, the best way to think about this is go home and play with water, take water, put it in put it in an ice cube tray, put it in the freezer, and then you have a solid. Okay, something with a solid is something with a definite shape and a definite volume. Okay, and then take the ice cubes out, put them in a glass, just let them sit there at room temperature. Just let them sit, okay? And then eventually, after a couple hours, depending on the temperature of your place of living, then the ice cubes are going to melt and form a liquid because the temperature went up, okay? The water molecules are moving, farther away from one another, okay? And liquids are variable in their shape and volume, okay? There isn't really a definite shape with the liquid, so you can manipulate them a little more. And then you put the water in a pot, stick it on the front burner of your stove, crank it up, and get the water boiling. Okay, then all of a sudden, the water molecules are moving even farther apart from one another with more energy, and then you develop a gas. Okay, a gas. And gases have very variable shapes and much more variable volumes than a liquid. Okay. So that's basically matter, okay? Anything that has mass and occupies space, okay? And as I mentioned before, the smallest units of matter are atoms, and atoms are what we find them in nature as elements, okay? You guys remember the periodic table of elements. The periodic table of elements has, all those different atoms and those atomic numbers that you see on there. And basically, these different elements are specific atoms that have specific properties to them. Okay. So each of them has their own, each element or atom has their own different physical properties, basically varying amounts of the subatomic particles that make them up. these elements and atoms can be detectable with our senses and can be measured. For example, carbon dioxide, which is in the it's carbon and two oxygens joined together, or oxygen. We can monitor that in the body. We can monitor hydrogen ion concentration in the body because that affects our pH. Okay, so these are states of matter that we can't see, but we physiologically can monitor these and know they're there. Okay, and they have various chemical properties that determine basically how they interact with other elements. How they're going to join together to form chemical bonds. Okay. Um, so bear in mind that atoms are, the smallest forms of matter. Okay. Um, Now, this is just some basic terminology that I kind of want you guys to be acquainted with. The major elements of humans the major ones are carbon, oxygen, hydrogen, and nitrogen. Okay, these make up the bulk of the mass of us humans. Okay, so carbon is a big structural one, and carbon is a unique atom because it has 4 valence electrons. I'll talk about what a valence electron is in a little bit. And it can bind with many different atoms and form lots of differently shaped molecules, which is good. Because that allows for a lot of diverse shapes in nature and in us. Okay, and then oxygen, we can use it for functional and structural purposes of molecules. We use it for metabolic purposes. Same thing with hydrogen and nitrogen. Okay, nitrogen you find on proteins. Okay, and we have a lot of proteins in the human body, and nitrogens are part of the building blocks of proteins, the amino acids. Okay, so these are the major elements of the body that make up the bulk of our body mass. And then we have what are called lesser elements. Okay, now, because we call them lesser because of the quantity that you find them in the human body. That doesn't, by calling them lesser, that does not make them low in importance. In fact, these are extremely important to have. Okay, I mean, you guys are all familiar with calcium. I mean, everyone tells you since you were a little kid, you should drink milk to have strong bones. Calcium does way more for us than just build up bones. Okay, I mean, your heart won't be able to operate properly without calcium. Okay, and if your calcium levels get too high or too low, you're going to see some pretty severe cardiac arrhythmias take place. Okay, muscles cannot contract. Skeletal muscles cannot contract without calcium. Okay, so calcium is very important for many different reasons. Magnesium we use to regulate smooth muscle, smooth muscle tension, and how constricted or dilated blood vessels are. Phosphorus is an important buffer and is used for ATP and structure. Sodium, potassium, extremely important electrolytes that we use to, use for nervous impulse conduction or muscle contractions or regulating body water levels. I mean, iron, sulfur, these are both extremely important as well in terms of structural components, structural components of amino acids or, hemoglobin and iron. Okay, these are, like I said, we just call these lesser elements because they are found in smaller amounts in the body. And from a nutrition standpoint, so we call these lesser elements, so that means that I mean, you do have to take in a fair amount of these, but not as much, as you would need carbon, oxygen, hydrogen. But basically, anything you eat is going to have high levels of carbon, hydrogen, oxygen, and nitrogen. Okay, well, the nitrogen may be variable because if you eat a lot of plants, you're going to have a harder time getting protein in your system. Okay, and then trace elements, again, another nutrition term. Trace elements means that these are just found in a very, very small quantities, less than 1% of our total body mass. Okay, so again, from a nutrition standpoint, that means you do not need to consume a lot of these. That doesn't mean, again, they're not important, but you just don't need to overload these in your system. And basically, these are used by enzymes to activate enzymes, and I'll talk more about these later on when we talk about enzymes. Okay, so now let's kind of jump into the atoms. Okay, now as mentioned earlier, atoms are the smallest units of matter. They are composed of 3 separate subatomic particles: protons, neutrons, and electrons. Okay, and then that's what makes, that's what makes different atoms different, is the amount of protons, neutrons, and electrons. Okay, so for example, we'll say black represents protons. Okay, we'll say red represents neutrons, and we'll say green represents electrons. Okay, so one thing to bear in mind is that protons have a positive charge. Okay, neutrons have no charge. Okay, and electrons have a net negative charge to them. Okay, so how many did I draw here? 1, 2, 3, 4, 5, 6. So typically there are so basically 5, 6. Now, typically there is an equal amount of protons and neutrons within what we call the nucleus of an atom, but this isn't always true. Some atoms have more neutrons than others, and those are what we call isotopes. Okay, so isotopes are atoms that just have an increased number of neutrons, and that won't affect the chemical properties of the atom all that much, or actually at all. What it's going to do is it's just going to increase its mass. It'll just make the atom heavier. Okay, so you've got protons and neutrons within the nucleus of an atom, and then you've got electrons found in these kind of these shells orbiting around the atom. Okay, and this is what really makes these atoms different, is the amount of protons compared to electrons, and neutrons as well. Okay, now electrons, what was I going to say? Electrons are important because these affect the chemical behavior of the atom itself. Okay, so basically, for starters, I'm drawing this right now. Okay, something to think about. So this atom has 6 protons, 6 electrons, and 6 neutrons. Okay, should draw this like this. Okay, so 6 of each. Okay, 1, 2, 3, 4, 5, 6. Okay, what would the net charge of this atom be? What would the net charge? What would be the electrical charge of this atom? There are 6 positively charged protons, and there are 6 electrons. Now, being the neutrons don't have a charge, these don't have an actual net charge, these won't really affect us at all. So being that there are 6 of each, there is no net charge. It's neutral. Okay, but let's say I take away, let's say I take away one of the electrons. Okay, so now this atom has 6 protons and only 5 electrons. Now this has a net positive charge. Okay, has a net positive charge. All right. Now, if I do the opposite, if I add, an extra electron, and there are 7, it'll have a net negative charge, okay? And you're going to see this from time to time, okay, amongst the different atoms, different amounts of protons to neutrons to electrons, okay? Now, I will say this, is that protons and neutrons stay within the nucleus, okay? They don't go anywhere, okay? You guys all know what happens when you split the nucleus of an atom, okay? You get something that looks a little bit like this. Okay, that's what we think about with nuclear weapons. Okay, so basically when it comes to atoms and themselves interacting with one another, okay, the electrons are what interact with other atoms, okay? And basically what happens, electrons are either released, they can be accepted, or they can be shared between other atoms, okay? And I'll talk about the different types of bonding in a little bit, but for all intents and purposes, just understand that protons and neutrons are found within the nucleus of an atom, and that protons have a positive charge, neutrons no charge, they're neutral, and that electrons are found kind of circulating around the nucleus of an atom. Now, one thing to keep in mind as well is that as you have these shells farther out from the nucleus of the atom, there's a lot less energy holding these electrons into the atom itself. Okay, so as a result, the farther out the electrons are, the easier they are to get rid of. Okay, so basically the outermost electrons are what we call valence electrons. Okay, and these valence electrons are going to be the ones that are going to be the first ones that interact with other atoms. These are the ones that are really going to do the chemical bonding or affect the chemical properties of the atom itself. Okay, so kind of keep that in mind about these atoms as well. Oh, and something I forgot to mention, I apologize about that, is when you look at the periodic table and you want to talk about the atomic number, basically what that is, that's the number of protons within the nucleus, okay? So, so if this has 6 protons, its atomic number would be 6. This would be the 6th element on the periodic table. That's all that is, okay? Now, there are, as I've already mentioned earlier, there are variations within atoms, and like I said, the variations lie in the ratio of protons to electrons. Okay. Now remember, basically, as I already kind of mentioned these, that there are some atoms that are positively charged, and there are some that are negatively charged. Okay, these are what we call ions, as mentioned above. Okay. Ions, basically, these behave differently due to the electrical charge of them. So basically, so remember, let's say if we're talking about a positively charged ion, okay, there are going to be more protons than electrons. Okay, and let's say the nucleus of this atom has, oh, I don't know, let's say the nucleus of this atom has 11 protons and 10 electrons. Okay, you know that's going to be positively charged. Okay, so we would write that. So let's say I'm just throwing these around here. Let's say we're talking about sodium. Okay, since sodium is missing 1 electron, there is 1 more positively charged subatomic particle than there are negative. We would write that as Na plus. Okay, that's how we would write that. All right, if it was missing 2, if there were 9, okay, we would write that as Na2 plus, but you're never going to see that. You're going to see that with calcium, though. Okay, because calcium's always written as Ca2 plus, magnesium, Mg2 plus. Okay, iron, depending on how you find it, could be Fe3 plus after we start to digest it. Fe2 plus. Okay, so what we're saying here are these calcium and magnesium ions are missing 2 electrons, so there are 2 more protons and electrons. This iron here, has, 3 more protons, so it has 3 more positively charged particles. Okay, so that's basically what ions are. Okay, and basically the positively charged ions are what we call cations, and the negatively charged ions are what we call anions. Okay. So cations and anions, okay? And one thing about these is that these ions are going to interact with water in a specific fashion. I'm going to talk a little more about that later on when I talk about the properties of water, okay? Something else to keep in mind as well about these more heavily charged elements, the heavier the charge on these, the harder they are to digest and absorb, okay? Iron is not easy to absorb, all right? That's why if you ever seen an iron supplement, those pills that people take, they're about this big, okay? So basically you try to get as much iron in your system as possible and hope some of it's getting in. Okay, same thing with calcium and magnesium. These are not easy elements to absorb. Okay, so the more neutral, the easier it is to work with in a sense. Okay, and as I mentioned earlier, isotopes, all these are just basically elements, or not ions, I'm sorry. These are elements that just have more neutrons. And basically all that's affected is their mass, okay? And you'll hear about isotopes in the clinical setting. There are some radioactive isotopes that are used for scanning purposes. You'll inject an isotope into somebody's blood, and then it'll go within a certain tissue, and then you'll run that person through a scan, and it's going to light up like a Christmas tree, and then you can see what's going on with that specific tissue or organ that isotope is found in. Okay, and plus these are used in research as well, but so these are the various different variations you can find within atoms, okay, ions and isotopes. All right. And then when atoms start to interact with one another, you start to form molecules and compounds. Okay. And most atoms combine with one another to form these, and there are just certain molecules and compounds that are much more abundant in us than others, and those are the ones I'm going to focus on, like carbs, fats, proteins, and nucleic acids. Now, molecules are basically two or more atoms bonded together, okay, whereas a compound are two or more different kinds of atoms bonded together, such as glucose. Okay, forget I put that there. Okay, so if you take two hydrogens and put them together, that would be molecules. Okay, or the six carbons linked together would be molecules, and then you start adding the okay, so that's molecules and compounds. Okay. Now, energy is the capacity to do work, okay, and the capacity to do work, okay. Now, we need to start talking about energy because we're going to start talking about chemical bonds, and we'll talk about metabolism later on in enzymes. But basically, where does energy come from within us? Energy comes from the chemical bonds that are found in between these atoms that are holding these atoms together, okay. So it does take a certain level of energy because remember, that depending on what type of bond it is we're talking about, these are electrons that are either being weakly donated or shared between two other atoms. Okay, so as a result, we can, if we break this chemical bond, all right, if I detach this chemical bond, then I release energy. Okay, so that's where we get energy from to do work at the cellular level. All right, and that's what we would consider potential energy. All right, and then there's kinetic energy. So basically, this potential energy is energy just sitting within here, and then if we break this chemical bond, and then we could use this energy to actually create work. Okay, create movement. All right, for example, the contraction of a muscle. If we start snapping apart, phosphates groups off of an ATP, okay, and we release the energy from that. I'll talk more about ATP later on. That's going to make a muscle contract, okay, cellular energy, all right. So, and the different types of energy that you're going to find in the body, those are two general classes. But the general types, again, I just mentioned chemical energy found within chemical bonds. So the bonds holding together sugars, the bonds that hold together fat, proteins, okay, whatever it is, if you break bonds, you're going to release a little bit of energy. Okay, some molecules just have a lot more chemical bonds and have a lot more energy holding the molecules together, so therefore you will yield more energy by breaking them apart, like adipose tissue and fat. Okay, this is our main form of stored energy. All right, and so fat, we can get a lot of energy out of fat. Okay, there's electrical energy, okay, and this happens when you move ions around and generate what's called an action potential. You generate an electrical difference across the cell membrane, okay, and you're going to learn more about this later on when I talk about membrane potential. All right, so I'm going to leave that be for now until I get to that specific topic. Okay. Mechanical energy, that's pretty obvious, movement. When I hear mechanics, I think movement or pressure. Okay, so basically this would be a muscle contraction. Okay, and as I mentioned earlier, if you break apart ATP or start snapping bonds, or separating atoms apart, the molecules apart, you can create movement. Okay, and then radiant energy, this is, what I think about here is photo energy, like the sun. Okay, photo energy or compressed air, that allows us to generate hearing. Okay, well, I suppose that's not electromagnetic. Okay, but so this is energy coming from the environment, to us that we use to create sensory, phenomena like hearing and vision. Okay, and as mentioned earlier, the different forms of energy are found within chemical bonds. Okay, and there are different kinds of chemical bonds out there, and one of them is an ionic bond. Okay, bonds that occur between cations and anions. Now remember, cations are positively charged, anions are negatively charged. Okay, now one thing to bear in mind about these is that they are very weak. Okay, these types of ionic bonds are very, very weak bonds. Okay, and they occur, but they occur between cations and anions. Okay, now let's think about this for a second. As mentioned before, a cation is an atom that has more protons than electrons. Okay, and in order for it to reach a neutral state, it's going to have to gain more electrons so the charges can even out. Okay, so as a result, cations are always, cations are going to be accepting electrons, whereas anions, being that they are negatively charged, they've got more electrons than protons. Okay, these are always going to be trying to donate electrons. Okay, so as a result, these two have something to gain from one another. Okay, in order for them to reach a neutral state, cations need to get rid of electrons. Anions need to get rid of electrons. Cations need to be accepting electrons. Okay, so as a result, these two interact with one another. Like sodium chloride, table salt. Okay, if you separate the two, sodium has a net positive charge and chlorine has a net negative charge. Okay, so this has one extra electron. This is missing an electron. So as a result, these two are going to interact with one another if you get them close enough to one another. Okay, and there will be a very weak chemical bond between these two. That's something to bear in mind about ionic bonds, is that they are relatively weak. Okay, they're easy to separate, and I'll talk about this later on, is that they're very easy to separate in water. Okay, so you find a lot of electrolytes bound together like this. Okay, and all right, and again, like I said, I'll go a little more into this in a little while. Okay. Covalent bonds. Okay, covalent bonds occur when electrons are shared between two atoms. Okay, when electrons are shared between two atoms. All right, now these are a lot stronger chemical bonds. Okay, covalent bonds are a lot stronger. So let's say we have two hydrogen atoms that are going to bond together. Now, covalent bonds are always represented by a line. So this line means this line between these two hydrogen atoms means there is one pair of electrons being shared. So let's kind of doodle this out here. So let's say this is a nucleus, this is a nucleus. Okay. So when I say one pair, if we're talking about a pair, that means there are two electrons involved. All right. So now these atoms are closely linked together, and they're sharing the energy. They're sharing these two electrons. Okay. Now these are a lot stronger of a chemical bond. All right, these are much stronger chemical bonds, so it takes more energy to snap these apart. Okay, so especially with big molecules like proteins that form a lot of covalent bonds, these are, that's why it takes so long to digest proteins. They're big, complex, and it takes more energy to separate these. Okay. So that is and I mean, it's physically and chemically impossible because hydrogens, don't have this many electrons. But let's say, if I draw two, if you see two lines like this in between atoms, then that means there are two pairs or four electrons being shared. Okay, if you see three, now there's 6 electrons or 3 pairs. Okay, and bear in mind, the more of these covalent bonds that are taking place, the stronger, the more strongly joined together those atoms are. So it's going to be a lot harder to break or separate that, those two atoms or the molecule. All right. So, for example, when we talk about DNA and when we talk about complementary base pairing later on, okay, which two are going to be harder to separate? Okay, obviously there's more energy holding, the cytosine and guanine together. So as a result, it's going to take more energy to snap all 3 of these bonds to separate those. Those apart. So if the molecule has, more of these triple bonded C's and G's, it's going to be harder to separate. Okay. And there are also two different types of covalent bonds. There are what are called polar and non-polar, okay? And what we're talking about here is basically how are the electrons distributed around the molecule, okay? Around the molecule or the compound. All right, so basically, for example, if we say something is let's go with non-polar. If we say something is non-polar, let's say this is just 1, 2, we would say that the electrons are evenly distributed around here. So there's an equal amount of electrons kind of flowing around this molecule here. Okay, there's an equal distribution of them around this. So as a result, there is no charge. It is neutral. Okay, non-polar. That's what we're saying here. We think charge, okay? If I say non-polar, no charge. Okay? And a very common non-polar substance we're going to be talking about are lipids or fats. Okay, so the opposite of polar then would be something that does carry a molecule or a compound that does carry a charge. Okay, and the most common polar molecule that we're going to talk about is going to be, oops, sorry about that. The most common polar molecule that we're going to be talking about is water. Okay, an oxygen with 2 hydrogens. Okay, being that oxygen has way more electrons, has 16 electrons, okay, versus hydrogens that only have 1, okay, there's going to be an unequal amount of electrons, between the poles of this molecule. So the side where there are going to be more electrons, i.e. the oxygen, okay, is going to carry a net negative charge. Okay. The side where there are less electrons and more protons, coincidentally, there's going to be a net positive charge. Okay, so basically, polar. When we say a molecule is polar, one side is negative, one side is positive. Okay, and this is and being that water, being the polarity that it carries is very interesting because this allows it to be a very universal solvent, and I'll talk more about this in a little while. Okay, but that's all we're talking about with polar and non-polar. Okay, polar, there is a charge due to an unequal amount of electrons and distribution of them in a molecule. Non-polar meaning there are. Okay, and one thing to keep in mind about this as well is that when we're talking about polar and non-polar, think about this: water and fat. Okay, how well do these two mix together? Okay, how well do these two mix together? Okay, for example, think about this. Have you ever put oil in a glass of water? Okay, how well did that oil stay in that glass of water? It didn't, right? They try to separate from one another. And that's because of the differences in the polarity between the two molecules. They separate from one another. Okay, there is no chemical bonding taking place. They have nothing to gain from one another. Okay. And this is important to know and understand then when we talk about, for example, when we get to the endocrine system, when we talk about steroids. Okay, steroid hormones. Steroid hormones are made out of fat. They're made out of cholesterol. Okay, and we circulate. So basically, think about this. If these are cholesterol-based molecules, how do we circulate them around in the body if our blood is mainly water? Okay, there is a way around this, but that's just something to think about. Okay. This is also important when it comes to determining how cells interact with their environments, okay? Because a cell membrane is made out of fat. Okay, cell membranes are made out of fat. Cell membranes are nonpolar. Well, steroid hormones are also nonpolar. So guess what? Steroid hormones can just move right across the cell membrane into the cell. All right? Whereas hormones that are made out of proteins are polar. They're charged. Uh-oh. So how in the world do these protein-based hormones, if they can't cross the cell membrane, how in the world do they activate activity within the cell? And that's something I'll discuss more in the next chapter, but that's why it's important to understand the differences between polarity and, polar and non-polar, okay? And last but not least, I just want to talk a little bit about hydrogen bonds. These are actually really cool bonds. Hydrogen bonds are really neat because these influence the shape. That eraser is really cool, isn't it? Okay. hydrogen bonds influence the shape of molecules. Okay, hydrogen bonds typically occur, between an oxygen or a nitrogen. Okay, so hydrogen bonds occur between oxygen or nitrogen. All right, and as a result, what happens is they make specific molecules kind of bend or twist or coil. All right, and as you're going to learn a little later when I talk about organic chemistry and biochemistry stuff, I'm going to draw you a protein here. Try not to be too mesmerized or amazed by how cool or interesting this looks. See, you've all probably drawn a protein at some point in time in your life and you didn't even know it. Okay, this would be a big protein. This is what we call a tertiary protein. But basically what happens as we make proteins, as we start to link together amino acids, okay, what's going to happen is they're eventually going to start to form hydrogen bonds with one another. Okay, so a hydrogen, one amino acid, is going to start to bond with a nitrogen or an oxygen and another amino acid, and it's going to make this chain start to bend and coil and twist. And then this protein is going to start to bend and coil and twist, and it's going to eventually take a globular shape. It's going to take on a specific shape. Now that's important because the shape of a protein is going to dictate its function. Okay, and if we start to alter the shape of the protein, that's what we would call denature. Or we would say we're denaturing the protein. Okay, so when you see the word denaturing, this is a word you're going to see from time to time. When we say we're denaturing something, we're changing its shape. Okay, and a specific term for the shape of a molecule would be conformation. Conformation. Sorry, I can't spell today. So, okay. So this protein has a specific conformation, okay? And if I stick a bunch of acid or mess with the temperature of the environment that these proteins are found in, I would disrupt these hydrogen bonds and I would start to denature the protein. I may make it uncoil or untwist. Or I may make the protein coil even tighter or twist together even tighter. But in the end, either of those scenarios are going to be bad because that's going to disrupt the function of the protein. Proteins have specific shapes. Okay. So keep that in mind about hydrogen bonds. There's a reason why DNA has the look that it has. Okay, that double helix look to it. That's about the best I've ever drawn that. Okay, because wherever you see the kink in the strand of the DNA, that's where a hydrogen bond has occurred. Okay, and between the strands of the DNA, and that's what makes it twist and coil, and that's good because that allows DNA to be a little more structurally stable, and also makes it a little easier for us to store it as well. Okay, so that's all hydrogen bonds are. These are relatively weak as well. These are relatively easy to disrupt, in the right environments. Okay, like I said, stomach acid is going to denature these proteins. All that acid in the stomach is going to make the protein uncoil, denature the protein. Okay, extreme enough temperature changes are going to be enough to denature the protein as well. Again, if it's too warm, remember we talked about this, proteins are going to uncoil. If it's too cold, they may coil tighter. All right, but these are the various types of chemical bonds you're going to find out there. So ionic bonds, whoa, sorry, I thought I had the marker still up. Okay, ionic bonds, relatively weak bonds. Hydrogen bonds, also relatively weak bonds. Covalent bonds, very strong. The strongest out of all of these are nonpolar due to the equal distribution of the electrons and the energy that's holding this molecule together. So these are going to be the hardest the hardest molecules to separate from one another. All right. So as you're looking through as you're watching this, you've got your book out in front of you. Make sure you're looking at the pictures in your book as I'm talking about all this as well. Okay. And then mixtures occur when we basically take certain molecules and put them in water. Okay. We take certain molecules and put them in water. All right. Now, one thing that's cool about water is that water is very, very polar, as I mentioned earlier. Okay. So, and being that water is so polar, being that one end is very negatively charged, One end is very positively charged. Okay, it can interact with basically anything that has a charge to it. Okay, so any molecule that is polar can basically interact with water. Okay, and so proteins, carbohydrates, ions, electrolytes, these all interact with water. They can be mixed in with water, whereas non-polar substances like lipids, fats, they cannot be. Okay, so as a result, so when I take a solution, that would be when I mix in a very small concentration of a certain, element. Let's say I put some ions in here. If you want to make a solution, go home, take a glass of water, put a little spoonful of sugar in there and stir it. Okay, put a little more in there and stir it until you start to see the water get a little cloudy. Okay, maybe it'll get a little cloudy because it's going to start to get saturated with whatever you're mixing in there. Okay, so solutions are basically water mixed with very small substances that dissolve very easily in there. Okay, that's all they are. Okay, so if you have a bunch of electrolytes, if you have a bunch of salt in there, that's a solution. All right. Colloids, what colloids are, these are gels. Okay, so basically, if you ever made Jello, you've made a colloid. Okay, because what you're talking about here, you're taking big molecules like proteins. Okay, proteins have a very strong attraction to water. Okay, proteins are unique because they're polar. They're unique in many ways because of how they mix with water, because of how they interact with, they can act as buffers, they can act as acids, they can act as bases. They're really dynamic molecules. Okay. But when you take proteins and you mix them in water, okay, what's going to happen are the water molecules are going to, you're not going to form a gel is not a solid. It's kind of in the middle between a solid and a liquid. Okay, and what's going to happen is the water molecules are going to start to get in between the I probably shouldn't use the same color for this. The water molecules are going to start to get in between the coils of the protein and interact with the polar aspects of the proteins. Okay, so as these water molecules get trapped in here, okay, the proteins are going to latch onto those very tightly. But obviously not all the water is going to get in between these coils here. There's going to be a little bit of water found outside of the proteins as well. Okay, that's why you don't get a true solid and you don't have a true liquid either. Okay, because you have a lot of water, because you do have water suspended in between the coils of the protein, but there's also enough outside to kind of make it nice and wiggly. Okay. And, basically when we're talking about colloids in the human body, we're talking about connective tissue. Okay, because when you take proteins, I mean, that's all connective tissue is. You have cells in the body that squirt out a bunch of proteins into your water compartments of the body, and then you form these gels. Okay, and these gels are relatively sticky. Okay, hence that's why we call them connective tissue, because they stick other tissues together. Okay, for example, your blood is a colloid. Okay. It's a connective tissue, because of all the plasma proteins in the blood. All right. You guys have seen blood before. You guys have all had a cut at some point in time. Blood doesn't just pour out of you like water. Blood oozes out of you because blood is so thick. Okay. Because of all the proteins mixed in the blood. All right. It gives blood that kind of sluggish nature to it. All right. Same thing with connective tissues in the body. So that's all a colloid is. And these molecules are big. These proteins just aren't going to freely move across cell membranes very easily or at all. Okay, so that's another thing to bear in mind. And suspensions, or what are often called emulsions, are basically when you start to separate water from whatever substance you're trying to mix with it. And probably one of the more common ones you're going to think about would be the fat in breast milk. That's probably the only one you're really going to deal with. Okay, but that's all those are. All right, so, and that's kind of what's neat about again the polarity of water is it can mix with so many things. And these are the different kinds of mixtures you're going to find in the human body. The two most common ones you're going to hear about are solutions and colloids. Okay, especially when we get to the chapter on histology, chapter 4, you're going to hear a lot about this when we talk about connective tissue. All right, so that's all mixtures are. And another thing to bear in mind, just some basic terminology, solute and solvent. Okay, so if I have a beaker of water, all right, if I have a beaker and I put some water in it, you know, the solute would be, well, the solute would be I'm sorry, not the solute, the solvent. Ugh, not with it today. The solvent would be the water, okay, and then the solute would be what I'm putting in the water to mix. Okay, so like as I mentioned earlier, if you're making Jello, okay, if you're making Jello, when you make that, you rip open the pack, you pour it in the water. Okay, you put all that protein and sugar in there, and then it mixes with the water. All right, if you made hot, it's winter time. If you want to make hot cocoa, okay, the water is the solvent. The solute would be the cocoa mix. Okay, so that's all those terms are: solute and solvent. Okay. And some differences between mixtures and compounds. With mixtures, there is no chemical bonding, okay? And they can be, and these can be easily separated just by straining or filtering or spinning in a centrifuge. Whereas compounds, okay, if you want to separate compounds or molecules apart, you actually have to break the chemical bonds. Okay, you actually have to snap the chemical bonds that are holding, so for example, if I have a sugar molecule, okay, and I want to separate this apart, I actually have to snap, the bonds between the carbons, the hydrogens, and the oxygens. Whereas with a mixture, due to the polarity of water, all right, due to the polarity of water, let's talk about this for a second. I didn't really get in depth to this like I should. All right, so if I have water here, so as I mentioned before, ionic bonds are very weak, and ionic bonds are easy to disassociate in water. All right, they're very easy to disassociate in water. So what happens? Let's say you put table salt in your water. All right, you're cooking, have a weird diet or strange taste, whatever it may be. Now remember, this is a weak chemical bond between a positively charged cation and a negatively charged anion. Okay, so if I put these in the water, what's going to happen is the water is going to interact with these, but it's only going to do it physically, not chemically. So now let's just look at the sodium here. Now remember, the negative pole of the water is the oxygen, the big oxygen atom, and the positive pole is is where you find the two hydrogen ions. Okay, so what's going to happen then so let's focus just on the salt, the sodium aspect of this. So what's going to happen, let's say I've got a sodium here, the water molecules are going to surround the sodium, so it's going to look like this. Whoa, whoops. Started that right, and I messed it all up. Okay. All right, so the negatively charged pole, the negatively charged portion of the water, is going to be facing the sodium. Okay, whereas in the same beaker, if we're looking at the Cl here, what you're gonna see is this. Okay. The positively charged pole is going to be facing the Cl. All right. So as a result, but like I said, you notice these aren't actually physically attaching to either one of these. The water molecules are just separating them. Okay. And now, and if you want to bring this to life, okay, take a bunch of salt and put it in water and then boil it. Okay. And then boil the water and then, boil it. And then when you have that little bit of residue left at the bottom of the pot or the pan or whatever it is you're using, okay, what's happening is you boil the water molecules started to move around, bang around, and eventually became a gas and evaporated out. Okay, and then as they evaporated out, as more and more water left the pot, there's less energy forcing the sodium chloride apart, so they start to come back together, and you form this salt residue at the bottom of the pot. Okay, if water actually physically and chemically bound to this, then it would take the sodium or the chloride with it, and there would be nothing left in there. Okay, so that's something to keep in mind as well. Why, and then that's also why it's easy to separate, these mixtures. Okay, and you'll see this a lot when you're trying to separate, blood, when you spin blood and things like that. All right. Okay, and then, to kind of change the shift to focus here a little bit, metabolism. Okay, metabolism is basically this, as I mentioned in the chapter 1 explanation, the sum of all chemical reactions. And there are two major types of aspects of metabolism that we talk about: anabolism and catabolism. Okay, when you see this word, a lot of people think anabolic. People tend to think about steroids right away, and it's not incorrect to think about that, because when you are, this aspect of, of our metabolism, anabolism would be forming chemical bonds between atoms. Okay, so you're building molecules. Okay, you're building something up, you're creating, not creating, so you're making something. Okay, so as a result, this is what we would call endergonic. Okay, because we are taking, remember, it takes energy to hold these two atoms together. Okay, and the energy that's holding these two together is stored within this covalent bond here, this chemical bond. Okay, so the opposite of that would be catabolism. Okay, catabolism would be the breaking of chemical bonds. Okay, and so if I snap this bond apart, then energy would be released, and then I would use that to perform work, movement, generate an action, whatever. Okay, so that's why I would say exergonic, releasing energy. Okay, and these are happening all the time. We're continuously forming chemical bonds. We're building, we're continuously making proteins. We're taking small amino acids, linking them together, 25 to 50,000 of them, and then they're coiling up and forming these big proteins. Okay, we're constantly taking these sugar molecules, okay, and snapping the bonds that are holding them together. Okay, so we can get the energy out of that to perform work at the cellular level. Okay, so we're always, building and breaking chemical bonds. That's metabolism. Okay, and like I said, regardless of whatever the type of chemical bond it is, whether we're talking hydrogen, ionic, or covalent, okay, that's our metabolism, making or breaking these chemical bonds. Okay, that's all metabolism is. All right. So as a result, when we're still talking, kind of keeping in with these chemical bonds, there are three major types of chemical reactions that take place. Okay, decomposition, synthesis, and exchange reactions. Okay. When it comes to decomposition, reactions, okay, this is relatively simple. I mean, so let's say, for example, we take, probably a good example of this would be digestion. Okay, digestion. All right, so let's say you eat a protein. Okay, proteins are just too stinking big to absorb. Okay, they're too big of a molecule to absorb as we take them in. Okay, and there are other reasons why we don't want to absorb whole proteins as well, but we'll just stick with the size thing. Okay, so you have this big molecule of 25 on average to 50,000 amino acids linked together. Amino acids are the building blocks of proteins. Okay, so if I want to get this protein into my system, and you guys all know you need to eat protein in your diet regardless of how you do it, whether you eat twigs and berries or you eat meat, okay, you have to get protein in your system. It's an essential part of staying alive. Okay, and so what we do then when we digest food is we snap this, we snap down this protein. Okay, so there are chemical bonds holding these 25 to 50,000 amino acids apart. We snap them. Okay, and then we form these individual amino acids. Now these are small enough to absorb into the bloodstream, and then we can put them to use. We can store them. We can use them to, we'll use them to, to build other proteins in the body that we need. Okay, plasma proteins, structural proteins, whatever. Okay, but the gist of this is we are taking a larger molecule and we are breaking it down into smaller parts, a smaller molecule. Okay, and like I said, digestion is the easiest example to think about that with. Or if you eat a plant, okay, if you eat vegetables, or fruits and vegetables, when they store carbohydrates, they store it as starch. Starch is just a very, very big complex chain of glucose. Okay, glucose molecules. All right, but you can't just absorb starch. It's too big. Okay, so as a result, when you eat these complex, carbohydrates, like, these big carbohydrate molecules, you have to individually snap the chemical bonds and then absorb the single sugar units into your bloodstream. Okay, that's a decomposition reaction, taking a big molecule, breaking it down into smaller parts. Okay, and then the opposite of that would be synthesis. Okay, a synthesis type reaction. And so, I mean, if you're thinking the opposite, well, this would be, so let's say we just talked about digesting proteins. Well, let's make a protein. Now we make proteins in our cells. Okay, when we make a protein, basically all we do is we find a little ribosome somewhere, and then we just start to link amino acids together. Okay, and as we link these amino acids together, we may form smaller chains, or we may link, some hormones are only, could be 11 amino acids long. Okay, small primary structures, or they could be big structural proteins that are as big as this. Okay, but in the end, even if I'm only linking together 3 amino acids, that's a synthesis type chemical reaction. I'm synthesizing something. I'm making a bigger molecule. I'm taking smaller molecules, joining them together to make a bigger one. Okay. So that's all that is. And then basically when it comes to, then the other major type of chemical reaction that occurs in the human body is what's called an exchange reaction. And this is probably the most common type of chemical reaction that's going to take place. Okay, an exchange reaction. So basically, if you want to write these out, so let's say we're going with decomposition. Okay, so I would write it like this, decomposition. I would go A, B, C, draw an arrow here, and then I would have A plus B plus C. So you guys kind of understand this. We have one big molecule here. That's what the letters A, B, C represent. Now this arrow, whenever you're looking at chemistry, you're looking through your book and your chapter, and you're seeing these arrows drawn. These arrows represent a chemical reaction. Okay, and these chemical reactions are driven by enzymes, which I'll talk about more specifically later on. Okay, because I want to talk more about the structure and nature of proteins before I kind of talk more to enzymes. Okay, but so basically there's a chemical reaction that takes place. We broke this apart, and now we've got smaller units, plain and simple. Okay, a decomposition reaction is this. A synthesis reaction we would write like this: A plus B plus C. Okay, so relatively simple. I take I'm out of it today. I take 3 smaller units, a chemical reaction occurs, and then bang, we have, this is a synthesis reaction, we now developed a bigger molecule. Okay, so here's how an exchange reaction would work. Okay, so what happened here was basically you can see what happened here. We started out; these 2 molecules came together. Okay, these 2 molecules, came in contact with a specific enzyme. A chemical reaction occurred where we separated the molecules apart, and then another chemical reaction took place. And what do you notice happened? The molecules exchange their parts. Okay, and this is primarily what happens in the body because this is just energy efficient. All right, we don't want to just break molecules apart and then get rid of parts when we could be reusing them, okay, to form other molecules. I mean, this is how we make stomach acid. We to buffer our pH of our body. Okay, these, many chemical reactions that take place are exchanged, but that's all these are. Okay, is when we come in contact. When different molecules come in contact with an enzyme, it's broken apart and the parts are exchanged. That's all that is. Okay, so then think about this. Which one of these would be endergonic? Which one of these would be anabolic? Anabolic, endergonic. Which one would you be forming chemical bonds and building the molecule? A synthesis reaction. Okay. That would be endergonic. Okay, whereas which one would be the exergonic chemical reaction? The catabolism. This right here. Okay, so those terms we just talked about. All right. And then this is kind of a combination of both. Okay, first we break the bonds apart. We catabolize these molecules, and then we build them back up. We anabolize them. Like I said, that's more energy efficient to do it that way. Okay. So again, a lot of terminology that goes with this. I'm going to leave the oxidation reduction alone until I talk about metabolism more in depth. And then basically this here, the factors that affect the rates of chemical reaction. Okay, now you have to remember that chemical reactions, when they take place, they're not as simple as, when you look in the books. Okay, I mean, you'll see chemical reactions written out like so. If we want to look at a common buffering system, HCO negative, blah, blah, blah. Okay, so it's not as simple as these just all line up and then all of a sudden they start to form together or break apart. Okay, basically chemical reactions are influenced by, because this kind of goes back to the Schrodinger's paradox that I was talking about a little while ago, or in chapter 1, is that chaos. I mean, this is all about chaos and molecular motion. Okay, molecules are always in motion. Molecules are always moving around. Okay, and molecules are going to eventually at some point in time bang into each other. Okay, and as they're randomly moving around and banging into each other, okay, the right ones may bang into each other and a chemical reaction may occur. Okay, enzymes, again, as I'll talk about later on, make this occur more efficiently. Okay, so basically, when we talk about factors that affect the rates of chemical reaction, what we're talking about, what are factors that will influence the random motion of these molecules? What will increase the chances of these molecules to move around and bang into each other more often and generate more chemical reactions? Okay, if we increase body temperature, that's going to increase, that's like saying we're increasing energy and we're increasing molecular motion. So as a result, if we increase body temperature, these molecules are going to move around a lot more. There's more chances of them banging into each other. There will be more chemical reactions taking place. Okay. If we have a lot of smaller particles, again, it's easier for smaller molecules. You might be thinking, well, they're bigger. They might bang into each other a lot more easily. You're not wrong to think that, but remember, this is about molecular motion, okay? The bigger the molecule, the harder it is for them to move, okay? So the smaller the particles, the smaller the molecules, the more motion is going to take place. Again, the more chances they're going to slam into each other and a chemical reaction is going to occur, okay? Increase concentration. Well, this is pretty obvious. If I just increase the amount of if I increase the amount of buffering hydrogen. So for example, this is what I just put up here. Let me rewrite this again. Okay, what this is here, this is a very common buffering system of the body. Okay, and I'm going to talk about pH a little later. Okay, but basically pH revolves around hydrogen ion concentration. So if I need to get my pH in check, if I'm becoming very acidic because my hydrogen ion concentration is getting really high, okay, if I want to get this under control, I'm going to retain and secrete more of this buffer, bicarbonate. Okay, so the more of this buffer that I keep in my system, or that I release into my system, the more the chances are this bicarbonate is going to bang into hydrogen ions and carry out the right chemical reaction to form this carbonic acid. Okay, and then I circulate this carbonic acid around. It's going to get to the lungs. It's going to run into a specific enzyme, and then you're going to form carbon dioxide and water. You breathe the carbon dioxide out, the water stays in you, all is good. Okay, but this is just a big part of how we regulate our pH in buffering, is that we want to increase the amount of this molecule we have here. All right, so I increase the chances of these two banging into each other, and then I make it easier for myself to regulate my pH. Okay, so that's all we're talking about there. And as I mentioned earlier, you know, one of the biggest factors, though, probably the biggest factor in the rates of chemical reactions would be enzymes. Okay, what are called biologic catalysts. Okay, enzymes really increase the speed and efficiency that chemical reactions occur within the body. But again, I don't want to get too in detail with enzymes until I talk more about the structure of proteins, which I'll talk about in another slideshow. So again, folks, if you have any, I know this was a lot of different terms, a lot of chemistry thrown at you. You know, read your chapter. If you have questions, don't hesitate to ask. But you know, again, I mean, just, you know, listen to this, draw this out, you know, follow along, use the images in your book, and you'll be all right. So now, a lot of this may not make sense right now. I mean, like I said, this is a lot of jargon, a lot of terminology. You know, I mean, I try to put some application to it, but I'll tell you what, if it doesn't 110% make sense right now, obviously I want you to continuously go back and read it and study it to make it make sense. But this isn't going to be the last time you're ever going to see this material, okay, on any of these topics and, you know, in this chapter, okay, even the ones I haven't covered yet. All right, so even though this may not make complete sense right now, once we start applying it and we start using it on specific concepts in physiology, you're going to be amazed at how familiar this is all going to seem to you. So like I said, don't get too down on yourself if this all doesn't click right away, right now. It will later on, okay? So be confident and just keep working at it. And like I said, if you have questions or need help, let me know. I'm always more than happy to help.